Thank you, Monica. Well, Dario gave a great introduction, very detailed analyses of the growth cycle. So I'm going to mostly focus on the last 60 days of ripening. So these series are great. We're going to have another one focusing on water relations. I'm sure you've been getting emails from service providers about the controversy of cavitation, so on and so forth. So I'll have the USDA scientist go through the water relations aspects in the grapevines in the next sessions on Focus on Physiology series. So we are mostly interested in wine grapes in the North Coast, although I do some work in table grapes as well in the San Joaquin Valley. So must composition, we are trying to optimize it from the points of sugars, color for lack of a better term, astringency as a tactile sensation, and also acidity. So most of the questions we get in extension or when we're writing proposals or writing papers is focusing on when do I need to pick these to express the varietal character of the cultivar that I'm working with. Well, everything has a reason, as Dario went through. A lot of these processes are transcriptomically regulated, and as viticulturists, we have limited tools at our disposal. We have not had a lot of developments in breeding of the grape varieties that we work with. We're working with about 8 cultivars in California. So how can you assess this ripening as you're going through these last 60 days of the growth of the plant? So Dario has shown this figure. Everyone has seen it a million times probably, but you have a berry formation and then berry ripening. So we usually assume that we have about 140 days to bring in the crop since flowering. So we have the double sigmoid growth curve where we have pericarp and cell division soon after setting. And soon after we start accumulating a lot of these compounds that we try to assess ripening with, such as proanthocyanidins, methoxypyrazines, and the concentration of malate. Dario did a great explanation of the conversion of malate to sugars in his presentation, so I won't go through that. But the last 40 days, last 60 days of this ripening is of importance to us because we tend to hang the fruit for a lot longer than maybe we should for some reason that I cannot really put a finger on. So starting on 0 degrees at flowering, you have the berry formation, and then you have this accumulation of malate, which then starts declining. When we look at the berry volume on the y-axis, following the double sigmoid growth curve, under optimal conditions, we have a vascular flow into the berry. However, after some point in time, we start seeing this shrinkage. And this shrinkage can be due to dehydration, overexposure, or a high heat gain due to changing in the climate, in the light spectra that we might receive on the clusters. When we look at the phenolics in the berry, for lack of a better term, for flavonoids, we assign some properties to these flavonoids. So you get the color from the flavonoids, you get the copigmentation from some of the other flavonoids like your flavonols. You get some tactile sensation from skin proanthocyanidins, and you get a taste sensation from seed proanthocyanidins. Of course, they do have health promoting effects, and they have important antioxidant properties. When we look at the international cultivars that we commonly see on the store shelves, you have Merlot, Cabernet Sauvignon, Malbec, Syrah, and Petit Syrah. These are the most common ones that we grow, and if you look at the changes in hue, in the intensity of color that you get from these cultivars, the varieties that we're known for, such as Merlot, Cabernet Sauvignon, or Malbec, they are not that intense in the color hue or the total anthocyanins that we might generate. Anthocyanins or flavonoids are produced through the phenylpropanoid pathway. You have this influx of phenylalanine. You end up with naringenin. It goes through some changes, and then it'll branch out into either proanthocyanidins or anthocyanidins here on the side. If we look at it more carefully, for the compounds as grape growers or buyers that we might be after, you have some constitution of the flavonol and anthocyanin profiles that we need to take care of as far as our basic knowledge is concerned. These compounds can be monosubstituted, disubstituted, or trisubstituted. The compounds circled here in the green are the flavonols, and this is the compound that can, these are the compounds that can co-pigment in the wine matrix with the anthocyanins. We have this monosubstituted compound called camphorol, and that's usually only a monosubstituted compound that we see, only under severe ultraviolet light stress and water stress. But everything is coming from the schimate pathway, and you have these two parallel downstream pathways. And I have put the abbreviations down here for the enzymes that are catalyzing these changes in these compounds. When we look at these, you have two parallel downstream pathways, and they all end up with these final compounds that are your color compounds, which are identified in the purple circle. Looking at the monosubstituted compounds, this is pretty straightforward. Flavonoid 3-hydroxylase changes dihydroxycamphorol into camphorol. When we look at the two parallel downstream pathways for the flavonols, you have the disubstituted flavonols such as quercetin, isoramnitin, and peonidin 3-glucoside. When we look on the other side of the pathway, these are catalyzed by flavonoid 3,5-hydroxylase. You have the trisubstituted flavonols. These are the myricetin, lyricitrin, delphinidin 3-glucoside, petunidin 3-glucoside, and melvidin 3-glucoside. We only see the glucosides in the berry skin, and quercetin, myricetin, these are usually going to be found in the wine matrix. We do not see these in the fruit. When we look at the color compounds, they are usually modulated by some hormonal process. They can also be disubstituted or trisubstituted, again following the same pathway. Two parallel downstream pathways are modulated by the same enzymes. You can end up with dihydroxylated anthocyanins and trihydroxylated anthocyanins. Usually, some sort of water stress or water deficits, whether it's a pre-veraison or post-veraison, will upregulate this side of the pathway to give you trihydroxylated anthocyanins. These are more stable. They do not oxidize as readily as the dihydroxylated compounds. There's a reason why these are shown in purple, because the hue you get from delphinidin-based or trisubstituted anthocyanins, they have this purplish, blackish hue. Cyanidin-based compounds, they're pinkish or somewhat reddish in hue. So these are readily oxidizable. But the effect of water deficits on flavonol profiles is not that well known, because we think that they are upregulated by light attenuation on the berry or the cluster. So you have disubstituted anthocyanins. Usually we don't have pelargonidins in the Vitis vinifera grapes. You have the disubstituted on the B ring of anthocyanins. They would be in the third and the fourth position of the B ring. And you have the trisubstituted anthocyanins. These are the delphinidin-based compounds. The substitution is going to be on the third, fourth, and fifth position on the B ring. When we look at the flavonol homologues, similar to cyanidins, the disubstituted ones would be quercetins. Monosubstituted flavonols would be similar to pelargonidins. These are camphoryls. We usually do not see these unless we have a severe stress. And we have the trisubstituted flavonols, which are similar to somewhat delphinidins. Why make a big deal about flavonols? We talk about mouthfeel mostly on pro-anthocyanidins. However, when you add quercetin 3-O-glucoside to white wine or quercetin 3-O-glucoside to red wine, they do have an impact on the wine mouthfeel, and it's quite significant. The intensity of astringency, persistence changes, but also bitterness changes as well. So astringency is a tactile sensation, bitterness is a taste sensation. So the balance between these two will be modulated by how reliable you can degrade the proanthocyanidins later on in the ripening season. So talking about proanthocyanidins, they are important due to their astringent properties. They have a role in long-term color stability. The grape-based proanthocyanidins, the monomers are catechin, epicatechin, epicatechin 3-O-gallate, and epigallocatechin. So there's a difference between seed versus skin proanthocyanidins. The skins contain epigallocatechin. They have a greater degree of polymerization and lower proportion of gallolated subunits. You get most of the gallolated subunit proanthocyanidins from the seed, and how you form reversible reactions with the salivary proteins in your mouth cavity as wine is swallowed or expectorated is mostly going to be modulated by EGC. So you have some changes in wine grapes after veraison, right? So you go through engustment, you have the influx of 6-carbon sugars, you start accumulating anthocyanins, proanthocyanins start degrading, and now you have a decline in malate and total acidity in this case. The figure on your left is changes A in berry volume, B is juice bricks, and C is weights of solutes per berry against days after flowering of a muscat cultivar. The different dates are days after flowering, so you have this different plasticity of the berry as you're going through ripening. And we see this in Oakville as well, where I'm stationed. The berry weight is manipulated very readily in these last 40 days of ripening by the amount of shading that you throw on these berries. Total soluble solids in California, there's not much home here to write about. However, as you change the amount of light or the amount of shading or the change in spectra of the light you throw onto these canopies or berries will affect the degradation or stability of anthocyanins in the cluster or in the berry. So there's a relationship between total soluble solids and color. The y-axis is total anthocyanins, milligrams per gram of skin dry weight. So this is a gravimetric measure. So in this figure, as you're going or accumulating more total soluble solids, your anthocyanins are declining. I'm proposing something else completely different here. When I put total soluble solids on the x-axis, looking at total anthocyanins here, it's increasing. So this is the phenomenon versus shriveling versus ripening in Cabernet Sauvignon. This is with a clone 7 on a 110R rootstock. So as I hang the fruit a lot longer on the vine, I am degrading this anthocyanin content quite readily. When I look at the total soluble solids versus total anthocyanins here, when I look at the dry weight, I seem to be increasing it here. So there's a fine balance between ripening and shriveling on how well we can extract these out of this matrix. Total acidity is declining as we ripen this. So as I mentioned, we have about 120 to 140 days to ripen these berries. There's an optimum stage where everything is at its optimum. Berries are still turgid. Then you start getting into loss of water, loss of acidity, loss of color, loss of astringency, aroma and flavor changes, and also the consistency or the viscosity changes in the berry. When we look at ripening stage versus color in a warmer climate, region 4, which might be accumulating close to about 2,000 growing degree days based on degrees Celsius. When we look at extractable anthocyanins in milligrams per liter of the volume, we see that these start declining quite readily after 23 degrees Brix. Unfortunately, they took the study only to about 26 degrees Brix. Most of our studies in Napa were taking them to about 32 Brix. We see this concentration of extractable anthocyanins decline by about 45 to 53% once we move away from 24 degrees Brix. Anytime we're hanging these a lot longer than 24 Brix, each degree Brix increases, resulting in about 7% yield loss per foot of row or per plant. When you look at ripening stage versus winemaking, in this case, they harvested grapes at different Brix levels, 20 Brix, 22 Brix, 23 Brix, 24, and 26 Brix. And these were fermented in a triplicate, and then they were put against a consumer panel. Without an exception, the consumer panel chose 23 or 24 degrees Brix fruit to make the wine. Because the consumer panel identified this ripening stage based on overall fruit intensity, purpleness of the wine color, viscosity, dark fruit flavors, and opacity of the red wine. So hanging the fruit a lot longer may not be all that great in the long run. So what would be the optimal maturity? Well, in the end, it's up to you, because everyone has different price points, different markets which they're chasing. As a grape grower, I would choose to pick them at this stage, or wait a little bit longer, get the dimples on them like a golf ball, or shrivel them up on the vine. As we're moving more towards mechanization, keep in mind that there's going to be about a 7% loss in mechanically harvested grapes. Are you going to be able to pick more grapes when they're more turgid than they're more raisin? They're going to take a lot more force to shake them off the vine in the long run. When you look at the evolution of flavorants in Cabernet Sauvignon, this work was done by Professor Besson Davis. So you have this vegetative flavors that turn into herbaceousness. Then you get into unripe fruit, green apple, citrus rind. You start getting into red fruit, cherry, strawberry, raspberry, cranberry. You get into black fruit, and then when you dehydrate them, quite a lot, you start getting into these prune, date, raisin flavors. So the optimum maturity, will have to fall in somewhere in here, and that's what we can try to manipulate based on our knowledge of the physiology. So the quest for a perfect maturity stage. In an ideal world, if we were doing this in a lab, it would be a nice bell curve. You'd have green berries on one side, perfectly plump berries in the middle, and some shriveled up berries on the other side. And they would be somewhat distributed like this. But wine is the result of a combination of all grapes. So the mean usually is around 24 bricks. And the mean is, some of these green berries at like 20 bricks, or some of these overripe berries right around like 28 bricks. Combining two bad things rarely make a good one. So you can have a synchronous ripening or asynchronous ripening in vineyard and plantation systems. Because vineyards are variable in space, and ripening varies temporally within a vineyard. So these four panels are all Cabernet Sauvignon, except for this Merlot. So this is in Lodi, Cabernet Sauvignon on high quad cordons. This is Sonoma, Cabernet Sauvignon on high quad cordons. This is Napa Cabernet Sauvignon, vertically shoot position. And this is Merlot on the central coast in Paso Robles on vertically shoot positioned cordons. In most of the cases, we can see somewhat of a normal distribution in these curves, except for this Sonoma field, where you have this almost bimodal distribution of ripening. You can remediate this ripening asynchrony by exogenous plant growth regulator applications. However, with seeded cultivars, it's somewhat difficult to balance all the auxins, cytokinins, and gibberellic acids, also with the facilitated transport of sugars in the berry. They have done this in Australia very reliably. However, in the end, the results were not all that great. They could, upregulate, Brix accumulation, make it more synchronous, but flavonoid accumulations were not all that great. There's also the role of seed to berry ratio on ripening asynchrony. DeLuke is a professor in Oregon, so they looked at a low seed to berry weight ratio and a high seed to berry weight ratio, looking at red, green, and pink berries. And the relationship is almost linear. As the seed to berry ratio increases, your sugar level, or it would be measured as degrees Brix, would decline. So you get different levels of variability. You can have it between plots. So this is a field in Lodi. So it's varying based on essentially soil properties. So these are ripening a lot faster than these ones. You can have levels of variability within a plot. This is a field in Sonoma, where the northeastern portion of the field is ripening a lot later than the southwestern portion of the vineyard. Or you can have levels of variability within a plant. Not all clusters will ripen at the same time or at the same rate. Or you can have levels of variability within a cluster. Not all the berries are ripening at the same rate or at the same turgidity. Then it becomes a logistical problem versus a physiological problem. Physiological problem, we can try to remedy by cultural problems, but a logistical problem is, more difficult to tackle. When we look at variability between plots, the causes can be genetic. You might have different clones, different cultivars, stuff on different rootstocks, different microclimate, different soil properties. This is going to be affected by terrain and also cultural practice. A management solution is harvest scheduling in this case, but variability within a plot is more difficult to tackle. The causes can be, also, of course, different clones again, soil, microclimate, terrain, and more importantly for us in the North Coast, diseases. So Dario Cantu went through his red blotch with the asynchronous ripening of red blotch infected berries, and it is a true problem. It's not just late ripening. The vine just does not make these compounds as it should. Variability within a plot can be handled by differential harvest, but no one wants to come back and pick this plot various times. Labor is very expensive, and this only works for continuous patterns. In a case such as this, where you have this almost binomial distribution, you have 1 or 0. You can like separate the harvest dates. It requires a certain knowledge of the plot. And strawberry picking approach in wine grapes is not really possible. You can do individual variable rate management. This requires great knowledge of the field structure, if there is any, and standardization. This may mean giving up on some yield for no reason, such as a shoot or fruit thinning. Then you have the variability within the plant. The causes, as I mentioned earlier, can be diseases, or it can be a low leaf to fruit ratio, or it can be sun exposure, row orientation. So Sarah Spade did this work about 15 years ago, where they compared east exposed, shaded, or west exposed berries, where they were manipulating berry mass, total soluble solids accumulation, titratable acidity, juice pH, and total color absorbance. So shaded berries, as we all know, are not going to accumulate a whole lot of color. They're going to have a somewhat low pH and a greater amount of total acidity. But what we're dealing with right now in these days with the changing of the climate in the north coast of California is we have almost too much exposure that's degrading this color. Again, individual variable rate management can be applied to deal with this variability within the plant. This would require a great knowledge of the field spatial structure, if there's any. Standardization of the leaf area to fruit ratios. This may mean giving up on sun yield again. Berry selection or sorting in the cellar. And there's less penalization if you tend to keep the grapes a lot longer on the vine, however. Variability within the cluster. You have diseases, low leaf area to fruit ratio, sun exposure, and uneven fruit set. Again, there can be a differential harvest. This will require several passes again, only for continuous patterns. Standardization. So leaf area to fruit ratio is what we manipulate regularly. First, initially with our pruning practices, then shoot thinning, then we do some leaf removal if there's a need. But again, these require great knowledge of the vineyard plot. So there's also the role of carbon fixation in berry ripening. The x-axis total soluble solids, the y-axis leaf area to crop weight ratio. They compared bilateral single canopy or quadrilateral cordon divided canopy. Depending on how these fruit are exposed, the optimum time to reach technological maturity would change on how much leaf area you have to hang. And this leaf area to crop weight ratio from Cleaver and Ducouzlian's review paper looked at just exposed leaf area, the functional leaf area that is photosynthesizing. So bilateral cordon single canopy, we're looking right around like 1.4 square meters of leaf area per unit of fruit. If we were to divide these canopies, this ratio goes down to about like 0.8 to 0.9. That's why we say usually in review lectures, this ratio needs to be between 0.8 to 1.4. Again, this will be dependent on what region you are working with. However, to grow these leaves, you need some sort of supplemental irrigation in California. The x-axis here is net carbon fixation rate. This is micromoles of carbon assimilated per square meter of leaf area per second. And the y-axis is total soluble solids. The control in this case received about 70% of crop evapotranspiration replacement. RDI treatment, regulated deficit irrigation treatment 1, received about 70% of crop evapotranspiration. Bud break to fruit set, then it was dropped down to 50% of crop evapotranspiration replacement, then ramped back up. RDI2 received 70% of crop evapotranspiration from fruit set to veraison. Then we dropped it to about 50% of crop evapotranspiration later on. While it doesn't take a genius to see that if you water them a lot more, you're going to increase net carbon assimilation and henceforth 6-carbon sugar accumulation in the berry. If we were to manipulate when we expose the berries to stress, whether during cell division, whether after lag phase, you're going to either change the number of cells that you set in these berries, or you're going to change the amount of engulfment, the volume expansion in these berries. Nevertheless, you can control ripening with manipulating your pruning practices. In this case, we looked at conventional hand pruning or minimal pruning. 2 consecutive years. Date of maturation to 22 bricks varied by about 20 days. Titratable acidity was quite different in both years. Juice pH was quite different in both years. Tartaric acid was different only in the initial year. The amount of malic acid at harvest was greater with minimally pruned vines, so that might indicate some of green flavor. However, total anthocyanins was greatest with minimally pruned vines, and anthocyanins per unit skin surface, however, was not a difference. So in this case, we can interpret this as a concentration effect, not necessarily a biosynthesis effect, but being able to harvest berries a lot later than conventional times might be of value as we're seeing these giant heat spikes that might be lasting 10 days or longer in the North Coast. So later ripening may decrease temperature at harvest. As I mentioned, we have about 120 days or 140 days to bring this harvest. So moving this to a cooler portion of the season might be beneficial if we can afford to hang this fruit a lot longer. I'm not saying we need to hang this to about 28 bricks, but somehow manipulating this leaf area to fruit ratio early in the season to make it ripen a little bit later. The red figure is average high in degrees Celsius at Oakville. It drops down quite readily, and average low in degrees Celsius. This usually occurs after 4:50 in the afternoon. So we have this break point after the 14th of September in Oakville that might be beneficial for us to change these leaf area to fruit ratios. The other thing we're seeing is elevated carbon dioxide levels. These are doubling the rate of photosynthesis in the grapevines. Majority of our vineyards are right next to highways that are traveled very frequently. When I started my career, the amount of ambient carbon dioxide available in a volume of cuvette was roughly around 350 parts per million. And right now, it's not uncommon to see this at like 415 to 420 parts per million of carbon dioxide in the ambient air. What the elevated amount of carbon dioxide is doing is upregulating photosynthesis very readily. So we're going from about like 10 micromoles of carbon assimilated to essentially doubling the amount of photosynthesis that's putting solutes back into the berry, because photosynthase have to go somewhere. You end up with a faster berry development that is associated with a higher carbon fixation. Days to veraison, days to maturity to roughly about 22 bricks is changing very rapidly. And fruit set plus 10 days, veraison, maturity, the amount of photosynthesis will more than likely decline as you go towards ripening. However, the rate at which we're photosynthesizing has changed greatly. And under a constant leaf area to fruit ratio, we are ripening a lot faster with this higher amount of photosynthesis that we are doing. This faster ripening is leading to a lower anthocyanin concentration, because glucose or glucogenesis kickstarts all this process. As we make a lot more sugar, we do not have time to make a lot of anthocyanin, or we might degrade it under higher ultraviolet light conditions. So is there any disadvantage to ripening too fast? Well, there is, and we're seeing this, we're hearing anecdotal evidence. We're playing with a lot of things with extended maceration to smooth out the tannins, so on and so forth. So we looked at 5 different polyethylene cloths with 80% transmissivity of visible light. So we looked at blue, pearl, aluminate, red, black, compared to an untreated control in Cabernet Sauvignon. What these nets actually do is change the ultraviolet light spectrum, and also the red to far red light ratio of the light that is hitting the berries or the clusters. When you look at the physical conditions of the berry, so here's open field, green is control, blue is blue, dotted one is pearl, black, red, aluminate. You can see that we almost have a 2x difference in the visible spectrum. But the more important stuff is happening here at the lower end in the ultraviolet light range. So we're dropping this with the black cloth, for example. Ultraviolet light B is only letting in about like 76% of ultraviolet light. White is filtering out about 30%. Silver is filtering out 60%. Red even more, so on and so forth. What this is doing is actually changing the temperature of the berry as it's going through the day. I won't bore you with the whole lot of details. These rows were facing northeast, southwest. So here's the morning side. Not much happening here with the shade cloths on there. Here's the afternoon side, where we start seeing a lot of separation between control and the amount of shade that's on these, that's hitting these berries. Well, to summarize this, northeast side, morning exposed, the y-axis in temperature in degrees Celsius. So it's from 6 in the morning till about 8 o'clock at night. So we have tracked this. So here's uncovered, shaded, and ambient. Not much is happening on the morning side. On the afternoon side, exposed berries, we are seeing a huge spike in the uncovered berries, 48 degrees Celsius, roughly about 128 degrees Fahrenheit. So they start heating up right around noon, get up to about 120 degrees Fahrenheit, stay there till about like 6 o'clock in the afternoon, roughly about 4 hours and 4 minutes. Most of these biological processes are going to be regressed or downgraded roughly around like 32 degrees Celsius. At 35 degrees Celsius, you start seeing cell death. Of course, I showed this slide a little bit earlier. The berries respond differently. So this is from 11th of July till about roughly 12th of September. Berry weight changes. And look at this aluminate here. This is that shiny shade cloth. It reflects back into the canopy, creates this giant heat gain. And this actually has killed a lot of our berries. Our berry size hits this maximum and then stays there and then declines a little bit with this black shade cloth. Total soluble solids is not affected that much. When we look at the juice pH, we see some differences. Again, we see very good response from the black shade cloth. And when we look at titratable acidity, the black shade cloth keeps the titratable acidity within parameters that we would like to see, so we don't have to do a lot of hand touches in the cellar. So here are the interactive means. This was a factorial setup study, but this is the figure I want to show you. In vines without nets, control, although we sprawled the shoots on the west side, on the afternoon side of the canopy, they did not make it to the end of the experiment. More importantly, we had this phenomenon with aluminate, where we lost about like 20% of the yield. Interesting thing that happened with the anthocyanin profiles, we are able to change the hydroxylation pattern. When you look at the control under grower practices, which received about 65% of crop evapotranspiration from fruit set to harvest, we have very little trihydroxylated composition in the anthocyanins. We are able to make this a little bit better if we are to put the blue shade cloth, combine it with a regulated deficit irrigation practice. But when we look at the kinetic development of anthocyanins in Cabernet Sauvignon on the west side of Oakville, we see that we are able to optimize our anthocyanin concentration. By the 30th of August, wait about a week to harvest this with the black shade cloth, we're still within our parameters. If we look at control, which is uncovered with green, this is dropping by about like 43%. So they're essentially bleached out in this case. Well, of course, we looked at some of these genes that might be upregulated, downregulated under ultraviolet light or water stresses that you might impose in your vineyards to finish out the crop. Total skin anthocyanins, we are going to upregulate them if we are to irrigate them well, right? You have more substrate. And you are able to increase them again if you are able to put on the correct amount of ultraviolet light. The amount of ultraviolet range that's hitting the earth has increased in the recent years. So here are the dye-substituted anthocyanins. These are the cyanidins, pinkish ones. Pinkish ones are going to increase a lot more if you throw more water on them. So the enzyme that upregulates is increased. You get the best of both worlds. However, if you are able to combine some sort of filtering to ultraviolet light and introduce some stress early in the season. So I'll summarize this. If I just increase the amount of total ultraviolet light, I end up with a situation like this. I get a lot more cyanidins than delphinidins. If I introduce a water deficit, I improved the composition of anthocyanins in the Cabernet Sauvignon berry. They're more bluish, blackish, if I just introduce some sort of deficit early in the season. If I'm able to, string out some ultraviolet light to about like 25% of the available, combine it with a water deficit, as we have seen with the black shade cloth, I completely change the color profile of the berry. You get this darkish, inkish color in the Cabernet Sauvignon berry at this level. Of course, anthocyanins are not the only thing in the grape berry. We make a big deal about the mouthfeel. So we looked at a mean degree of polymerization at harvest with these different shade cloths. These did not influence it greatly or at all. However, seed mean degree of polymerization changed. So blue shade cloth with a regulated deficit irrigation reduced the seed mean degree of polymerization. So did black and red under a sustained deficit irrigation or the grower practice. So you can manipulate these very easily based on transcriptomic regulation, knowing some basic physiology, and then following this through the season. How can you explain the flavonol anthocyanin profile behavior with transcript levels? Well, as we're changing the light amounts with these shade cloths or the water amounts with irrigation treatments, light amounts are affecting the total flavonols, anthocyanins, methylated flavonols, methylated anthocyanins, quercetins, and cyanidins. You get the effect of water on the two parallel downstream pathways on the trihydroxylated side. And if you are able to combine the UV with water deficits, you're going to get a lot better response from delphinidin-based compounds. You're going to get them more darker, inkier, and they will not bleach out. Anyways, so we have this new experiment at the Oakville station. We are looking at these leaf area to fruit mass ratio manipulations. We're looking at 100% leaf area with 100% fruit load. So leaf area to fruit ratio will be 1, drop down to 0.5, 0.25 versus 50% crop load, 25% crop load. So we have these 9 treatments that we are going to track for the flavonoid expression of these profiles through the next 3 years with a PhD student to give a better understanding of how these leafing, pruning treatments work. We have about 8 to 9 years of experience with mechanization in the Central Valley. However, this is just creeping up to the North Coast, and unfortunately, we do not have recommendations for the North Coast as labor prices keep increasing. But our goal is not to look at it from the labor perspective, but when we started these studies, we said, oh, we could save costs, but we have seen more consistent yields with a lot more improved berry composition, not just from the total anthocyanins or pro-anthocyanins point of view, but the composition shifts towards something the winemakers would like to see. So, my soapbox, after 23 bricks, berries experience dramatic changes, better extraction of phenolic compounds, but also we get into a degradation of these compounds. The ripening stage of berries follow a bell-shaped distribution. These can be spread, so I do not necessarily care about the average. The impact of ripening asynchrony on wine quality deserves more attention. More detailed studies are needed on this. And knowing your vineyards, your own research, or the an impartial party may be the greatest asset to solve some of these problems. So with that, I'll quit. Thank you. Oh, yeah.
The acoustics are so good up here, I feel like I should sing my question, but I'm
Oh, the light is shining in my face.
I've got a two-part question. First off, on the minimal pruning, what does that consist of?
Minimal pruning is usually when you coppice about like a foot of the previous season's growth and let the vine sprawl out.
So like 5 buds or 6 buds, something like that? Or?
No, we left essentially about 30 buds in this case. Let the vine self-regulate.
Per cane?
Per cane.
Okay, and then second on the shade cloth, do you think there's a way to somehow dye the kaolinite or some kind of an applicable material?
Yes. Yeah, very good question. So we've done a lot of work with kaolinite, but repeated applications are somewhat necessary. What we have seen with kaolinite or these expanded clay applications is we stop or we arrest photosynthesis. And as we throw more water onto these vines during the hotter part of the season, we get into this vicious vegetative cycle. But regardless of this vegetative cycle, we are able to accumulate more total anthocyanins, but we're not able to degrade, in this case, or catabolize methoxypyrazines. So it's a give and take. So with kaolinites, we were seeing about 11 to 15-day reapplications.
Would a focused treatment, I mean, if you were just really targeting the fruit zone, do you think help to avoid the photosynthesis issue?
Well, that's difficult to say because not all the leaves are photosynthesizing at the same rate and at the same time. So the distal leaves or the adaxial leaves to the cluster are not photosynthesizing as much as the ones that are closer to the cluster. So they age, but they do catch up. But the problem we have seen was so the same enzyme catalyzes both methoxypyrazines and anthocyanins, and it encodes for the same gene. So once you start upregulating anthocyanins, you have already upregulated methoxypyrazines a lot earlier, so they do not burn up, for lack of a better term, later on in the season. But if that's the market you're going after with this green character with bell pepper flavor, that might be a good thing.
What level of exposure does the control treatment have?
The question is what level of exposure did the control have? All the vines had about 15% of PAR exposure. If you look at the photosynthetically active radiation spectra, they all had 15%. So it was you have the canopy, right? The canopy is filtering out some light, and then we put the shade cloth on it. So when we put the septometer on it, 85% of the light was being transmitted through it. So it was fairly open. But what is changing is the amount of ultraviolet light that's falling onto these clusters. So in the visible spectrum, everything was the same. The thing that we are able to figure out is that it's like those black balls that they put into the open air reservoirs in Los Angeles to stop them from evaporating. The black shade cloth absorbed all the heat and just let the light filter through. The other ones, they do have some changes in the spectra, but they are not as intense or as effective as the black shade cloth. In an ideal world, I'd put the shade cloths on top of the vineyard and be done with it like the table grape or blueberry growers, but I think they're going to kick me out of Napa if I suggest that.
Any other questions?
You put
Any other comment that you noted the increases in CO2 levels in your lifetime or in your industry?
I mean, increasing by 75 parts per million. I started in 1997 to now. 75 parts per million is, you know, quite a bit in my lifetime, and I'm not dead yet, so. So I think it's something to you know, watch out for, and with these you know, heat spikes, constant 100 days with you know, very little diurnal variation that we're seeing, even in Oakville, it's something to you know, watch out for, because the photosynthase have to go somewhere. They're either going to the shoot tips, or they're either going to go to the clusters or to the berries. Since we do about 10 to 11 hand passes in North Coast vineyards, they don't have shoot tips to go to. They go to the berries. We generate these you know, little balls of sugar where the anthocyanins degrade very readily, because our vineyards are way too exposed in this case. We maintain these vineyards at about you know, 1.7 leaf layers before the cluster. That's why you know, we were seeing the you know, lower light levels in these things. But even with that, there's about 43% degradation in anthocyanins with no change in sugar accumulation. So it's something to worry about for me.
So shade cloth versus not thinning your leaf or leafing your canopy. I mean, is it really just about the shade, or is it about that black shade cloth?
It is really about the shade. So we've done quite a few leaf removal trials, mechanical, hand, what have you, on both sides of the row, timing. In procumbent canopies and canopies that sprawl, you do see benefits of leaf removal. But in vertically trained canopies, under this climate, we might not see the benefits that are purported to be, you know, seen. However, you know, leaf removal post fruit set prior to veraison does change the proanthocyanin composition. It will give you more astringency, whereas pre-bloom leaf removal will give you more low molecular weight flavonoids like anthocyanins, flavonols, or monomers of proanthocyanidins. But again, you know, this is in the case that you have you know, 4 to 5 leaf layers before you get to the fruit. In that case, that might be needed. In a year such as this, where we still have about like 85% of plant water available in the top 3 feet of the soil, some canopy manipulation is going to be necessary to bring things into balance. But I don't know what that you know, level is going to be until we start getting into bloom.